So let us quickly take a look at what Konzo disease is about.
Konzo cripples the victim, which leads to many difficulties in managing normal life. All reported cases were from poor rural communities where the staple food is cassava.
What we're doing here is we're using this precision breeding tool, gene editing, to remove the cyanide content from the cassava. My name is Jess Lyons. I'm an assistant project scientist at the University of California, Berkeley.
My name is Michael Gomez. I'm a postdoctoral scientist at the Innovative Genomics Institute at UC Berkeley. Cassava is a starchy root crop. This crop goes by multiple names, such as yucca, tapioca, manioc.
Tapioca is made from cassava. Gluten-free bread will often contain cassava flour.
People may know it in their boba tea, that you can extract the starch from those roots in areas where the soil is poor or facing extreme drought. Cassava is still standing.
One important factor about growing and eating cassava is that it contains the precursor of cyanide, and cyanide is a neurotoxin.
Toxic molecules that, when ingested, can have an impact on our nervous system.
So even in the context where you might think that the cassava has been properly processed, it may not have been actually sufficiently processed. And many people are probably getting cyanide exposure who don't know it.
So in the developed world, we have access to a great variety of food, including protein. And it seems that protein is quite important in being able to break down that cyanide within our bodies.
But in places where cassava is a staple food, what that means is people may be having cassava as part of every meal.
Ultimately, that long exposure to that cyanide within the plant, in combination with that lack of protein in the diet, can lead to neurological damage.
And that can range from neurodevelopmental deficits to various paralytic diseases. So there's a very dramatic example of this called Konzo, where people can get paralyzed.
Konzo is a disease that has affected hundreds of thousands of people. It's also irreversible. And so if we can retain that nutrient value, reduce cyanide that's present, that would be fantastic for this crop.
What we're doing here at Berkeley at the Innovative Genomics Institute is we're using this precision breeding tool, gene editing, using CRISPR-Cas9, to remove the cyanide content from the cassava.
So through traditional breeding, we would cross plants and propagate them and hope that we could find that variation from plant to plant in a particular trait of interest.
The thing is, when you do that cross, you're throwing all of their traits up in the air, and they don't all land the same way in the offspring.
With the emergence of CRISPR, TALENs, and zinc finger nucleases, those make up this next generation breeding technologies. And with those, we could take work that would have taken many years and much effort and shrink that down to be much faster and easier to do.
So CRISPR-Cas9 is a tool that we use for gene editing. The way that we're using this gene editing technology in the context of cassava and cyanide is that we've chosen some genes that are required for the cyanogenesis pathway, for making the cyanide precursor, and we've targeted them with CRISPR-Cas9, and then we've chosen cassava plants that have disrupted versions of those genes so that the pathway can't function, so that it can't make the cyanide. I really enjoy working on African crops. I'm very drawn in by the problems and the opportunities to have an impact.
To think ahead to putting it out there and really alleviating a lot of that suffering that's out there. That is the dream. That is what we want to work toward.